So to conclude our day, our final question is our research Q&A, where you get the opportunity to ask your burning questions directly to our panel. Welcoming back to the stage, we have Professor Graeme Black, Kate Arkell, and two of our PhD students, Chloe and Gabriel, who will be joining us online, and Hasina had to leave. We were also welcoming Sergouniotis, Honorary Consultant in Ophthalmology and Genetics at the Manchester Royal Eye Hospital. Let's give them a big round of applause.
So, have we got any questions? Anybody wants to put their hand up? I've got a couple. Oh.
We got one at the front on the left-hand side in the light blue T-shirt.
Hi, my name is Marcus here. I've met Professor Black a few times, actually. But I was just wondering about the potential treatment for the X-linked RP. Would that be more of a preventative measure, or is it like a treatment in a way?
Yeah, so I'll start with that one, but I recognize I'm not the only one that knows about X-linked RP. But at the moment, the approach that's being taken by a number of companies is just a gene therapy. So much the same as for RPE65, albeit with a different gene, of course. And that's to say replacing the faulty gene to support the retina. I think we don't know. One of the trials is going to report, I predict, in March or April of next year, which will be a phase 3 trial, and that will be right towards the sort of the business end of approval of a therapy. The challenge there with X-linked RP is that you're by no means the only one that we know with it. And as I said, there's, we've diagnosed from a lab point of view about 1,800. I'm going to talk over the chiming. There's about 1,800 individuals. And Panos and I have talked a lot about this, but trying to work out how you deliver a therapy for so many people is going to require quite some thought, I think. And so at the same time, it's a little bit of a be careful what you wish for. We all want a treatment, but we've also got to be able to deliver the treatment to as many people in as equitable a way as possible. And so we're spending a lot of time thinking about that as well. I hope that answers your question. Now, the gentleman at the back who asked a question before, he's going to ask another question. Honestly. He's actually works for Retina UK. It's all right. He's monitoring the online ones, I predict.
Team privilege, Graeme. It's fantastic. So we've got a number of questions that are coming from people at home. So for anybody at home, please do put your questions in the Q&A. So this one is from Sandra. She asks, where can I get a list of the 300 retinal conditions to check whether my condition is on there? Progressive degenerative myopia. I have previously asked my GP for a referral, but he has no idea about this. Where can I get more information for my GP? Kate might talk about unlocked genetics as well with this one.
And so this is the point that I look at Panos and say, over to you.
How kind. Yeah, again, it's a little bit difficult to talk about specific situations because we don't have all the context. Progressive degenerative myopia, it's something that, it's a term that obviously we are familiar. We don't see them as much in our inherited retinal disease clinics because many times they're seen in the broader retina clinics, more general medical retina clinics. I would start with a specialist in a hospital, so just to see a retina specialist in a hospital and take it from there. The direct answer to where can I see the list of genes, there are lots of websites, but what Graeme was referring to when he was talking about 3 different ones, he was talking about 300 different ones, 300 different genetic causes. And the first thing to establish here is, are we looking into a genetic condition or it's a more complex environment plus gene situation? So we'll just start from a retina specialist just to see where we are.
Yeah.
Don't be shy.
I've got some. There's somebody there.
There's a
Following up from the last question, it seems to be very difficult to get a referral in the Manchester area for genetic testing. My partner's been trying for at least 12 years, seen various people at the eye hospital and his own GP, but nothing seems to have transpired. Is there a sort of a blockage in the system?
So the answer is no and yes, in that I've never come across that situation, and I'm sorry that you have. And generally, getting a referral via a GP into the genetics department, the Centre for Genomic Medicine at the Manchester University Foundation Trust, is a I mean, it's not a commonly trodden path, but it's a well-trodden path. So that ought to be plausible, and I don't understand why that hasn't happened, because it's accepted that it should. That having been said, you know, having promoted the NHS as being something which adopts novel therapies and is doing a lot, we can't pretend at the moment that there aren't some difficulties, and we have more referrals than we have doctors, and we have more doctors than we have space to house them. And so we are, to some degree, you know, there's more of you collectively coming at us than there are of us. And so there is no question that we have waiting lists for new patients, although we're pretty good with those, and we have follow-up waiting lists which are longer than we would choose. But that's a different question to the one that you're talking about. And insisting that your GP refers you to us, assuming you've got an inherited eye condition, but the truth is an ophthalmologist could do it as well, is entirely justified.
I've got some that have been put in the box during the day as well, and came in to us previously before today. So I'll just go in with one of these, which is something we get asked quite often. What's the latest advice on diet in general for supporting retinal health? Either of you, panels?
I can go first. Yeah, so diet is important. It's important for lots of things, and the usual things that doctors would say about diet, and I can imagine people being a bit cynical about it. So in terms of the evidence specifically for slowing down the degeneration in inherited retinal disorders, the evidence is not the strongest, but there is, I think, enough there to suggest that having a healthy diet with lots of green leafy vegetables, like what is described on the Retina UK website and leaflet, is something that is likely to be beneficial. If you don't do it, does that mean it's going to go faster? Well, we're not 100% sure. I don't think that anyone has. It's a difficult study to do if you think about it. Monitor people's diets and then just see, is it going faster compared to someone else? So the evidence is not very robust, but it certainly does no harm having a healthy diet, avoiding smoking, and avoiding exposure to bright sunlight. So, you know, just wear sunglasses if you somewhere where it's very, very.
You are talking in Manchester, though.
Yeah, that's. It's a rather sunny day though today. Yeah, I mean, but
I know, remarkable.
Yeah.
I was disappointed that Manchester let itself down by not delivering the rain. Yeah. Sorry, carry on. Yeah.
Yeah, so those are the three kind of lifestyle modifications we'd recommend.
And what about vitamin supplementation?
To answer that?
Yeah, I can answer that, but that sort of comes alongside diet, doesn't it? And again, I think The only thing to say is that there are conditions for which vitamin supplementation, for example, ABCA4 that we heard about earlier on, that's nodding behind me, I think, evidence to suggest that taking vitamin supplementation isn't a good idea. And so I generally don't advise it myself.
Can I continue on this point, or shall we move to the next one? Sorry, this is in the wave. Yeah, so in terms of supplementation, in the US, there has been a bit of a trend to recommend taking some vitamin supplements, the same thing that I would recommend for AMD. We don't do that here, just a balanced, healthy diet. We know that taking vitamin A supplements in ABC4 retinopathy, Stargardt disease, is not advisable. It's something that you shouldn't do. So in general, we wouldn't recommend supplements, and in Stargardt disease, we would just say don't, more specifically as well.
We've got
a couple nearer the front.
Hi, my name is June. My brother and I developed RP, X-linked RP, but we were in our mid to late 30s. Is that usual? Because I thought that, the retinitis pigmentosa affected males and coming down the female line. In my case, it started with juvenile macular dystrophy, so I had no central vision, and then I went on to develop RP. I was once told it could have become like this because of trauma. I don't know if you have any thoughts on that.
Would you like to?
Yes, so that's actually the type of RP that I'm investigating in that where you have the macular dystrophy first and the cones tend to degenerate before the rods. It's largely to do with the type of mutation you have and in what gene. And as to why, we don't know. Hopefully I will be able to find out. And when it comes to X-linked, whilst yes, it tends to affect males more. That's because males have an X and a Y chromosome, and the Y chromosome is very small and lacks a lot of genes, so you don't have two copies of the gene, whereas in females you'll have two copies of the gene. However, if both copies do have a mutation in a gene, you will still be affected.
So the only thing to say is that I think sometimes people say that the X-linked form, the sort of the loose way of describing is that it affects boys. And the truth is that I think in many families, female carriers do have symptoms, and in many cases can have quite significant symptoms. And generally, the boys are affected somewhat more severely, and perhaps at an earlier age, although that's not an invariant rule either. And so I think your family is not unique by any manner of means. The role of trauma in causing that, well, clearly trauma can do lots of different things, and we tend to look for explanations for things. But I think in this case, I would more likely look for an explanation in your genes and in the way that they're expressing in the retina than in something that happened maybe in your childhood or in your early life. And so I would tend to go down that route.
Shall we go for a question from
the
home?
Questions are coming quicker.
They are. If we go for a question from our online audience.
Yeah, so we have one that's come in. So myself and my two daughters have been genetically tested in around 2006 at Moorfields and have X-linked RP. How do we check if we're on the list so we can be contacted if trials are successful?
So I'm going to go for this one. Because the trials are moving forward and because the number of people is so large, I've actually been going through those lists. And it happens that for most of the patients with X-linked RP, not all by any means, and certainly not since 2019, we were the only lab that did it. And so I'm endeavouring to ensure that we create a list. I have, that I know who's on it, I do, and if there is success, I'll be contacting the clinicians involved. That having been said, Moorfields in particular are very thorough about maintaining their own lists and their own details. And so in this specific instance, I would expect with about a 95% likelihood that there is a list. I have it.
I can just add to that, Moorfields also have their own research database. You can, even if you're not a Moorfields patient anymore, you can join their research registry.
Yeah, although the list that I'm talking about isn't a research registry.
Okay.
Yeah, that's a clinical diagnostic. There's a difference there.
Okay. If you want to cover all bases, you can join that as well. If you Google research opportunities at Moorfields, you'll get to where you can register there online. But there's obviously some different places that could come from. Judy.
Hi, so my little boy was diagnosed with X-linked retinoschisis a few months ago, and I've really struggled to find out a lot of information about that condition, just with it being one of the rarer disorders. I just wondered if there is any research going on in that area at all, or anything I should be looking at.
Yeah, so X-linked retinoschisis is a condition we recognize, we see lots of families. I think it's a little bit, it's probably best to have a chat after, if you don't mind, and usually that's what I do. So I run a pediatric inherited retinal disease clinic, and I see people and I just give them the information and the research and so on and so forth. There isn't any active research on that, but it is one of the, it's been one of the main foci for the past 10 years. So there's lots of things that have been done and there's lots of plans, but at this stage, it's a very variable thing as well. That's why I'm a little bit reluctant to start discussing something. We can maybe chat after if you don't mind.
Hi. Yeah, thanks for talking today as well. I've wanted to ask this question for a while, so I know now I finally got the chance. I'm going to garble my words. But so there are therapies that for specific genes here, and obviously as time goes on, you foresee there may be more and more genes that there will be an established therapy for. And so in terms of the generalization of that approach to be able to get, now we can get this gene, now we can get the gene. Is there a pathway in the research towards where we get to a place where we can look at your gene and then almost a therapy can be developed for that gene, reasonably quickly because of the way the process has been distributed and shared? Or am I wrong, and it's the case that process is, for every gene, it's such a specific process and it's very difficult to generalize? So what is the situation in that respect?
Yeah, probably we'll both do this, but if I kick off, I think that's a really important question. At the moment, we're very much in the early stages here, aren't we? And so we've learned that we can replace genes in some circumstances. And we're learning to do it for one gene, and then we're moving out, for example, to the gene that causes X-linked RP or choroideremia, and there are other ones that are gene-specific replacement approaches. And the difficulty with that is that we need to go through all of the ethics and the safety questions in each time, and that's, more than anything else, time-consuming and very costly. And so at some point, we hope that we'll get to a point where actually once the therapy is there, we can just slot in cassettes. But I think the other approach is that we know that there may be lots of different causes, but the consequence of all of those causes sets off, if you like, it's a bit like those domino races that you see where you knock one over and another and another and another. once you've upset a photoreceptor, the upset photoreceptor has a certain biological or biochemical response that causes it to be unhappy. And so the question is, can we find some way of targeting that response rather than just replacing the initial cause? Because then we could take something where it wouldn't matter quite so much which gene, but we could develop something that was, if not universal, then a little bit more universal. And there are clinical trials for those that are beginning. They're in the early phase, looking, for example, at factors that promote nerve cells from not being unhappy or to promote them living a bit longer. And I think that something which doesn't worry so much about the cause but looks at the consequences is something that we really do want to move towards. I'll pass on to Panos just to carry on.
Yeah, so I think you covered most of it. Just two things there. One, people have started working on these gene agnostic approaches, as we call them, where it's not just for one specific gene, but it would work for a group of genes. But the question you asked is crucial. It takes a long time to do these clinical trials, and do we need to do that for every single gene? I think when it comes to gene replacement, like RP65 or all these things that are happening, we probably will. The key thing is what would need to happen for gene editing. So things like what Gabriel has been working, prime editing. If you find a way to edit, to correct one thing, can you just change the little thing, the little molecule, and then let it do it without needing to go through the trial for every single thing? So I don't know if Gabriel has any thoughts or insights on that, or I could continue as well.
Yeah,
But.
yeah. So I mean, it's very complicated question because there are a lot of different changes in the DNA. And for example, you could look in one location and that locus would be great for using the base editors that you mentioned before, but it might be terrible for using the prime editors. So it's very specific where the changes are. And like with Stargardt disease, you have over 2,000 different possibilities. So it will be really, we can optimize the systems to work better and we can try and test, but I think we'll still have that kind of rate limiting step where we have to look for the specific mutation, for the specific locus, what's the best thing that works. But something that I think we can improve on is later on, how do we deliver once this therapy has been created, like once we have the tools, because we have the adeno-associated viruses, but we could also use a messenger RNA, we could use nanoparticles, and I think that also should be a very big focus, because if we have that worked out in the downstream process, we know this is accepted by the immune system, we don't have like reactions, there are not that much side effects, that will definitely shorten the span for the generation of the gene therapy. So we have like a lot of very high intensive period of actually optimizing the gene therapy, but then later on it could be quicker. So I think we should definitely focus on that as well. I don't know if that makes sense.
Thank you. Thank you. Should we go at the back?
Back to online, or yeah, Matt, do you want to?
Yeah, this one kind of segues into what we've just been speaking about. It's around drug availability or treatment availability. So, assuming the successful completion of a phase 3 clinical trial, if it's successful, how long will it take for that treatment to be made available to people on the NHS through NICE?
Okay. That's a really difficult one, but if we look at what's been happening on the screen previously, one of the sponsors for this is a company called Johnson & Johnson, that are one of the groups that are working on that. And I think one doesn't want to promote any single company, but all of these companies are aware that there is an approval process. They are working very hard to develop the evidence to support approval, to foreshorten that as much as possible. And of course, there is already experience of gene therapies in the eye and other systems. And so in terms of developing the health economic evidence, the epidemiological evidence, the clinical evidence, and working with patient groups and patients to understand what the patient need is. And so there's no question that this is a complex process, and it's one that involves, either medicines agencies in the UK, MHRA, in Europe, or in the States. But the process is gone through in advance to some degree, so that it can be foreshortened. As to how long it takes, again, to some degree, it's a little bit of the I'll be with you in 5 minutes. It's longer than you would choose, but shorter than you would fear.
Just wanted to expand that. I think it's very intuitive, let's say, to think like, okay, phase 3 trial worked, can I have it now? So phase 3 is the final stage of when you're assessing something. But the question there is, okay, imagine this. So you say, I'm going to do a trial, and I want to save 3 photoreceptors over a year, and that trial works, and now I charge this medicine $2 billion per person. So that was a successful trial, but then it needs to go through some authorization process. Is it impactful to the individual? Does it alter the quality of life? And is it cost effective? And that's actually a major hurdle. It's quite a high bar there that needs to go. So a successful phase 3 trial is a great start, but then there's this process of approval that needs to go through the FDA in the US, in the MHRA, and so on and so forth. So it's a very important question, and it's a space that actually we're very keen to understand a little bit better. And actually, it's something that what perhaps has been overlooked was what has an impact on quality of life. And I think that's why lots of the trials, although they're successful, then they're not getting approved because the impact, it doesn't change something that matters to the individual. So if you get like a little bit more field, it doesn't change much. So I think that's one area that needs to be researched more.
And just to come back one more, in case anyone I don't know if there are any cynics in the room, but the cynicism that says, and of course the UK will be the slowest system, turns out not to be the case. And actually the streamlining of the system in the UK is recognised, even post-Brexit, not to be the popular myth that, of course, everything's bad in the UK.
And just to expand that, the economics is that these companies, they usually want FDA approval to make their product viable, because even if you get, so they really need to have approval from everyone to be able to go ahead.
Can I ask a question that's come on a card? We had a couple of questions actually submitted around children of people living with inherited sight loss who are concerned for their children. And I wondered, to generalise that a little bit, could one of you explain how genetic counselling can fit in to the process and the value that genetic counselling can add, perhaps especially for families that have an unclear genetic diagnosis or haven't had an identified genetic diagnosis? And how, if people are concerned for their children, perhaps a chat with the genetic counsellor can help with that.
I can just make a very quick point and pass to Graeme. I think genetic condition plus uncertainty, that's what genetic counsellors do. So they're very good at navigating that space and providing some more information and empowering. So I think it's a very important thing, input actually to an individual's care. So Graeme, I don't know if you want to.
Yeah, and I think So the process of genetic counselling, and both of us grew up as ophthalmologists, and then we trained in genetics afterwards, and the enlightenment that you get in terms of understanding about communication, about working with families, about taking time, giving time, answering questions is something that I think has been really powerful in my own education. And so working, generally ophthalmologists are keen to think about the two eyes in front of them, aren't they? The ones that sit on the slit lamp. But actually what genetic counsellors are doing is thinking about the wider family and how you think about working with parents and a child in an inclusive way. And that means working with uncertainty, providing information where it's possible, and working over long time periods. We work with families that we may have seen for 20, 30 years. We may see multiple generations of those families as well. And so I think the only thing I can say is that my own belief is that, fundamental though therapies are, working with genetic counsellors is something that's going to be absolutely crucial here.
And just to if people want a referral for genetic counselling, a GP can actually do that.
Well, and that goes back to the question at the back about, I haven't managed to get a referral. Genetic testing has been set up, and it is recognised that if you've got a genetic condition, genetic testing is broadly available, but it would be available alongside genetic counselling. And so referral into genetic services, and there's one in Liverpool, one in Manchester, one in Sheffield, one in Leeds, one in Newcastle, etc., Nottingham. All of those centres will have an ability to provide genetic counselling for inherited eye conditions.
Okay, thank you. Judy.
Oh, got one of them. Hi, it's Stephen Golden. Hi, Professor Black.
Hey.
Hope you're well. I have a rod cone dystrophy, RP2 gene, no useful vision now. A couple of years ago, I was listening to our conference online, and I heard a Professor Hodgkinson, I think her name was, who was talking about her pet project, which was the RP2 gene, and mentioning about growing an artificial retina. Was I dreaming?
Alison Hardcastle talking.
Yeah, I thought it was. And indeed, we heard about growing artificial retinas today, really, and organoids. And so this is a way that we can use multiple tissues in 3 dimensions to study the consequence of gene faults, to understand why a gene fault upsets the retina, and the mechanisms behind that can vary. So they were using artificial retinas in Petri dishes to study the retina. What they weren't trying to do was to grow a retina that they were going to put in anyone's eye, however. That's not what they were doing.
I want to add here a little bit. So we have an understanding about how the retina develops, but unfortunately, we don't know in much detail. So even the protocols that we are using, we are seeing a lot of variability in between different batches, in between different protocols. So really there is a big need for research that would help us understand how each individual cell type really specializes so that we can build these complex structures that would have all of the different cell types. There are people who are working on different cell replacement therapies, like cell patches, but also that again poses the question, if you bring a patch of photoreceptors, how do they communicate with the other cells in the environment? How do they communicate with the glial cells that are now supposed to relay that information into the brain? So it's a very exciting and interesting area of research. And we have learned a lot, but there is still yet a lot more on the way to be explored.
Thank you, Gabriel. We've got two over here.
Hi, my name is Matthew Albert. I'm an American and researcher working in San Francisco. I've got a comment and a question. First, I just want to congratulate everyone for this afternoon. It's been a sensational session, and I would like to reinforce, Professor Black, your comment about how well you're doing in the UK. I'm sitting here envious about the healthcare system and the opportunity to drive genetic-based therapies for people with inherited retinal disease. And there's incredible opportunity here. My question is whether there's thinking either by Retina UK or maybe some of the work any of you are involved with around establishing pre-competitive consortia with academia and industries such as Johnson & Johnson to be able to better serve the population once these new therapies are approved.
Yeah, and just quickly to say that both Panos and I work in an NIHR-funded biomedical research center. We've developed a vision-related theme within that looks at phenotyping and diagnostics, and we work alongside a therapeutics theme. And in that context, working closely with pharmaceutical companies, small, medium-sized enterprises, to try to look at the ways most efficiently to move forward is exactly what we feel is necessary. And the UK has a health system, which means that aggregating data is possible, that following large numbers of patients prospectively is possible, and we do have an integrated system. And so the question is, can we offer that as a way of promoting research with organizations exactly as you suggest? And were it possible for us to do that with companies such as yourself based in Silicon Valley rather than Alderley Edge, we would be delighted. Particularly for the collaborative meetings, which we would insist on being in your...
Never been to one of these, Graeme.
Yeah, Alderley Edge is very nice, you understand.
We've got, Yvette's microphone.
How are you doing, guys? So it's been really interesting hearing about the biology of inherent sight loss and the kind of interventions that exist to intervene on this biology to cure sight loss or make it better. I was just going to ask if you got any reflections on kind of psychological adaptation to sight loss when there's no kind of biological intervention which exists. I understand that you guys aren't psychologists, so I was thinking maybe it's just kind of reflections with in your time in like clinical practice, any experiences with people that you think are like better adapted, or any factors that you think are helping them or things like that.
Well, we'll both try that. I'll try one, which is that I think working with families and individuals has been an enormous privilege, and I think I've learned a great deal and been inspired by, quite a lot of the people in the room, actually, at the way that they do adapt. We spend time with individuals. We've talked about genetic counselling. I think that's also something that helps. We work with psychologists. We've got a psychologist based in our department, Rachel Faulkner, who also works with individuals. So are there different people? I think everyone's got their own way of coming to terms with vision loss. And if we can support people through that process, it's one of the things that I think we try to do, really.
Yeah, just to expand on that, I think taking a step back, I think about that a lot. You know, I think I just see that in a simple way, so genes plus environment. And like really one thing I would like to focus is how do we create an optimal environment for each genetically unique individual. So I have family members, close family members with sight loss, and I just remember as a young kid, you know, seeing how transformational audiobooks were to this family member, you know, for him, because suddenly, you know, just something changed for him, and you know, you see all these things about these new devices, the impact iPads had. I see lots of children, and I see how much the educational interventions they can just allow them to flourish, and I quite like the vision that Retina UK has about this focus on flourishing, and that's not just only about focusing on the gene, replacing the gene, getting a few more letters on the chart. I think it's just a broader concept. So yeah, I do think about it, and I think we can do better in thinking about there is the individual, there's the tumor, but there's also the environment, and how can this be optimized with the help of technology or other aspects. But as a department, I think we do that okay, especially the pediatric service, because we have this low vision assessments, and we have our eye clinic liaison officers and a psychologist, and we try to put it all together.
And I think it really goes back to what was talked about this morning as well, that value of community and peer support as well, potentially being really valuable. Okay, time's up, I'm afraid. Thank you all very much. Did you have something to add?
Well, and I think right back at you. You know, I think all of us on the stage are actually very lucky to be working in this field, and so for your time listening to us, and for the thanks that you give, our thanks in reflection. Thank you.
Thank you.
Thank you.
Thank you, and thank you all for coming so much today. Before I hand over to Paula for some final thoughts, I just have a few logistics. Firstly, for the person that bidded on the dragon outside, he is there to collect for you. Mark and Caroline will be at the front and the registration desk if you'd like to collect. And then secondly, to our volunteers in the room, after Paula is finished, can the volunteers please remain seated before we move on to the evening, so that we can manage those logistics before heading over to our celebration. And then finally, I would now like to hand over to Paula McGrath for some final thoughts on the day. Thank you.
Thank you, Lauren. Before you go, I'd just like to take a moment to thank you all for joining us today. Whether you're here in Manchester or watching us from the comfort of your own home, you have made this a tremendous day. I hope you would agree that it's been truly engaging and enriching. We've had insightful and informative discussions and inspiring presentations. I know I speak on behalf of the whole team at Retina UK when we say it's been an absolute pleasure to host you here today. It's been great to hear the buzz in the room, the connections being made. That's what today's about, to give you the opportunity to meet other people. It really is our favourite day of the year. Speakers and panellists, you've been fantastic. You've enlightened us, and thank you for taking time out of your diaries, especially on a Saturday, to share your valuable knowledge and experiences with us all. I must thank our sponsors, Johnson & Johnson and Ocuvision. As a charity that's reliant on voluntary donations to put on this conference, their support and generosity is greatly appreciated. Also to our wonderful BSL interpreters, they have worked so hard to ensure that this is a truly inclusive event. And of course, to all of our staff and our wonderful volunteers, your hard work, your meticulous planning, and your good humour have made this event possible. You should feel very proud today, Team Retina UK. Retina UK is dedicated and committed to serving you, our community, and your feedback is genuinely the lifeblood of all that we do. Please do make your thoughts known from today. You'll be receiving a feedback form. I hope as today has demonstrated, we're a charity that gets stuff done. As mentioned earlier, we rely on information that we gain from you through our insight studies to determine our future activity and to genuinely understand how we make the biggest difference for you. This survey will be repeated again in spring 2025. You'll receive it in Look Forward. So please do tell us what you think. Tell us about your experiences, your hopes and your aspirations for the future, and we will do our best to support you to achieve those. I hope you leave us feeling more positive and more optimistic. We're here for you and your families when you need us. We're here to support you to live your best life in whatever way you choose to, and to support you to make informed choices along the way. And we're driving forward research to find causes and treatments. We look forward to seeing you all again at any one of our online employer support group meetings, our face-to-face meetings, our webinars, our future conferences. In the meantime, please spread the word about Retina UK and what you've learned today. And for those here in Manchester, have a safe journey home. Thank you.